Chapter 11 is all about Mendel and the gene idea, and this is kind of a fun chapter. If you enjoy the idea of genetics, then this is the introduction to genetics. Mendel was a guy who's kind of known as being the father of heredity and the beginning of how things inherited from parent to offspring. And he was a monk who bred these garden peas, and he was able to select for certain colored flowered peas and the type of pea that it produced. And he bred them and discovered that there were ratios that would occur in their offspring with different traits. And from that, it started Mendelian law that we'll kind of get into talking about in this chapter. So Mendel probably chose to work with peas. We can never know because it was a long time ago, because there aren't a whole lot of varieties within the different characters that they have, but there are enough that you can determine some sort of you can see a pattern when you're breeding different traits. So he used characters, which were flower color, and they had different variants. So they were purple, white, and there wasn't a whole lot in between. And the characters, the flower color was character, but the variants, the colors that they produced were called traits. And he could control the mating between those two plants. And so he would artificially um, move the male pollen into the female part of the flower, produce offspring, and see what those look like. And another thing is that with these plants, they grew relatively quickly. So it's not like breeding um, it's not like breeding humans or even dogs. You see the offspring much faster, and so you're able to do multiple experiments in a relatively short period of time, gather enough data to see what outcome you get. And so he chose to track only characters that occurred in two distinct alternative forms. So it's either a white flower or a pink flower, and either a wrinkly pea or a smooth pea. And so he used varieties that were true breeding, plants that produce offspring with the same variety when they self-pollinate. So in a typical experiment, he would mate two contrasting true breeding varieties. So he bred white with purple, and he called that a hybridization. So what was produced from that was a hybrid. The initial parents, the pure white and the pure purple, he called that the P generation. And then the first offspring that produced from that P generation was called F1. And then if F1 were allowed to breed with another variety, their offspring was F2, and so on and so forth. So when Mendel crossed contrasting true breeding white and purple pea plants, all of the F1 hybrids were purple. And I'm going to show you a picture of this because just saying it makes no sense inside of your brain right now. When he crossed the F1 hybrid, so that next generation, many of the following generation had purple flowers, but some also had white. And as a result, he discovered a ratio of 3-to-1. This 3-to-1 ratio is really important. Get that in your mind, that there's always a 3-to-1 ratio when you're contrasting one trait, one character between true breeding parents. So we have our first generation. We have purple flowers and white flowers. And we call this true breeding parents because when purple breeds with purple, they'll only always produce purple. So he figured this out way a long time ago, right? So he bred a purple flower with a white flower, and the first offspring was called our F1 generation. And obviously, it maintained a color trait of only one of the parents, but it's got to have some of the genetic information from both of the parents. So when you take this F1 generation, where it's considered a hybrid, what he found was that their offspring, if you breed this purple flower with another purple flower, like another offspring from these guys, so siblings maybe, make that be a flower. What he found was that in the next generation, the F2 generation, he always had a 3-to-1 ratio. He always had 3 purple to 1 white. What this meant was that, and we'll get into this, what this meant was that the purple is the stronger gene. It will show up. And that white for lack of a better term, is the weaker gene. So he reasoned that in F1 plants, the heritable factor for white flowers was hidden or masked in the presence of the purple flower factor. So it was hidden. He called the purple flower dominant, and that's a really important term. So I said it was stronger. It is a dominant trait. It dominates over that white flower. And the white flower is the recessive trait. It's hidden. It doesn't show up until there's no dominant trait that's in the genes. The factor for white flowers was not diluted or destroyed because it appeared in the F2 generation. It was just hidden. A shadow went over it, and the purple always shows up first. Mendel observed the same pattern of inheritance in 6 other pea plant characters, each represented by only 2 traits. So what Mendel called a heritable factor is what we call a gene. He knew that this color was inherited by parents, but we call those genes. And so these are a bunch of results that he had. So he looked at flower color, purple versus white. There's no pink or anything that occurs in between. He also looked at their seed color. There was yellow versus green. He found a 3-to-1 ratio. He found seed shape, round versus wrinkled, pod shape, pod color, the flower position, and then even the stem length. And in every situation, his ratio was not exactly a 3-to-1 every single time, but it was basically a 3-to-1. And so he produced multiple experiments, replicated these experiments. You can see how many times. Sometimes he did it 5,000 times, 5,000 to, or nearly 5,500 to 1,800, multiple times, and always came up with this ratio, which means that it was a true scientific study. If he did it one time, it could be an anomaly. If he did it even just 100 times, it may not necessarily have given the same results. Mendel developed a model to explain this 3-to-1 inheritance pattern that he observed in that F2 offspring. And 4 related concepts make up this model that you guys need to be aware of. First, alternative versions of genes account for variations in inherited characters. Not all genes are exact. That's why we do not look identical. That's why dogs don't look identical. That's why even plants don't look identical every single time. There are alternative versions to genes, and that accounts for variations in the population. So in his experiment, the gene for flower color in pea plants exists in 2 versions. He had purple and he had white. And so these alternative versions, purple versus white, were called alleles. So you have a gene that has genetics. On your gene, there are things called allele. Your gene is made up of multiple alleles, millions sometimes of alleles. These are all different characters that can represent themselves in the way that you look: skin color, eye color, shape of your eyebrows, type of hair that you have, height, if you are predisposition to have diabetes or any other disease. All these alleles make up who we are and why we're so incredibly unique. Each gene resides at a specific locus on a specific chromosome. And I want to get to a picture of that, but I don't actually see it. So you guys are going to have to look that up in your book. The second thing that Mendel's experiments found was that for each character, an organism inherits 2 alleles, one from each parent. So we will inherit 2, if we're just looking at something really simple like curly versus straight hair. Dad has straight hair and mom has curly hair, and they only have straight hair genes, and dad only has curly hair genes. You're going to take 1 from each parent. He made this deduction without really knowing, which was amazing. He didn't know that there was an existence of chromosomes. This is way before that. So he was a really amazing person to put all of this information together without actually knowing the groundwork or the building blocks of it. 2 alleles at a particular locus might be identical, like in the true breeding plants, or they might differ. And we'll get into that in a little bit. That's called homozygous and heterozygous. The third thing that Mendel's experiment showed us is that if 2 alleles at a locus differ, then one, which is the dominant allele, determines how you appear. And the recessive allele has no noticeable effect on what you look like. So in the flower color example, that first generation from those true breeding purple flowers and true breeding white flowers, that first generation, they were all purple because the allele for that trait is dominant. It's the one that shows up. It is the appearance that you see. And then the fourth and final thing that his experiment showed us is this law of segregation. Law of segregation is really important. You see it in bold there, so make sure that you know it. The two alleles for a heritable character separate during the gamete formation and end up in different gametes. So when you are when you're a male and you're making sperm and you're a female and you're making gametes, you have, as a parent, you have two sets of this gene, right? You got one from your mom, you got one from your dad. Well, it has to split up when we make eggs. We talked about this in meiosis, right? It has to you cannot keep doubling and doubling and doubling chromosomes. So as it splits up, you end up it's all a mix. So all of your eggs, if you're a female, have a variety, an infinite variety of genetic information, a mix mash of mom's information and dad's information mixed together. And so during the law of segregation, these two alleles will separate and end up in different gametes. And it's no telling if you pass on your dad's information to your offspring or your mom's. So the egg and the sperm gets only one of the two alleles that are present in an organism. This segregation of alleles corresponds to the distribution of homologous chromosomes during gametes in meiosis. So Mendel's segregation model, he found that 3-to-1 ratio always in that F2 generation of his crosses. Always, always, always. So if you're given a question on the exam and it asks you, you have you're the P1 generation, you have a P1 generation of purple and white flowers. The F1 generation, what is the ratio of purple to white? Or any other character, and you are told what is dominant and what is recessive, your answer will always, 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 always be 3-to-1 if you're looking at one single character like color. Possible combinations of sperm and egg can be shown using the Punnett square to predict the results of genetic crosses between individuals, and we'll show you what a Punnett square is. It's a really cool genetic formulation that we use all the time to estimate and predict how things are going to turn out. So we use a capital letter to represent a dominant allele and then a lowercase letter that represents a recessive allele. So with the purple flower, big P is purple, little p is white. Use the same color because otherwise, or the same, we use the same color, the same letter each time, because if you did a P and a W, you'd get way too confused. So in this example, this right here is our square, Punnett square. So up here we have our true breeding parents. Big P, it is dominant, homozygous. Homozygous means it's the same thing. I think that term is going to come up a little bit later in this presentation. And then this one is recessive homozygous, right? It has only 2 little p's to give and only 2 big P's. So big P and little p are all they can give you. And so the F1 generation, the first generation from our parent generation, has to be a big P, a little p. Half of the time, it's a 50-50 chance whether it gives its offspring a big P or a little p, right? Because that's all it has to give. And so what you do is if, I don't remember, there's always a term. Oh yeah, males are over here, and then females are down here. So our female F1 parent can only give a big P and a little p. Same thing with the dad, big P and a little p. So you put them in this little square. We have 4 options, 4 things that can arise from this, 4 possibilities. And basically what you do is if it's big P, you bring it down, little p and you bring it down. So it's, I'm sorry, big P and big P. There you go. Down here, little p and big P. Same thing here, big P and little p. Always do the dominant up ahead of the recessive just because you don't see the recessive, so it'll end up confusing you. It's always hidden behind. And then in this case, they both give a little p and a little p. And if you look, since the big P is the dominant, it's going to show up purple. You've got 1 purple, 2 purple, 3 purple, and 1 white. So you have our 3-to-1 ratio. If you can go into your course tools and you can do some practicing on making Punnett squared, that will make all of this information so much easier for you. It's practice, 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 because all it is math problems. The tutoring lab is always open for you also to get some additional help. In addition to that, the homework, dynamic study modules, and the quizzes will have questions to help you get through this so that you get it enough times that hopefully it becomes a really simple concept for you. An organism with 2 identical alleles for a character is said to be homozygous. Homo means the same thing. Homo is the same. So it's homozygous for an allele. Homozygous was, these guys are homozygous, right? This is homozygous, and this is homozygous dominant, and this is homozygous recessive. An organism that has 2 different alleles for a gene is heterozygous. Hetero means different, so it has the dominant and the recessive trait. Unlike homozygous, heterozygotes are not true breeding. It's a hybrid. It's got a mix of it. So because of the effects of dominant and recessive alleles, an organism's traits do not always reveal its genetic composition. So we have 2 different things that we talk about. Phenotype is the physical appearance, and our genotype is our genetic makeup. So when you had, with these pea plants, when you had homozygous dominant and heterozygous plants, they have the same phenotype, they have the same physical appearance, but they have very different genotypes, right? They look the same, but their genetics are different. So if we looked at something in humans, let's say widow's peak. Widow's peak is a dominant trait. So if you have the widow's peak, you could be heterozygous or you could be homozygous dominant. What you know is that you're not homozygous recessive. Same trait, but you could have different genotypes. Okay, so in this case, we have more genotype options here than we have phenotypes. Phenotype is just how you look. That's where we got that 3-to-1 ratio. Genotype is a bit different. It's what's involved in your genetics. How can we tell the genotype of an individual with a dominant phenotype? Well, we don't. We have to do a test cross. So they could either be homozygous dominant or heterozygous, right? All they have to have is the dominant gene. They can have 2 or they can have 1. The answer to figure out how you tell the genotype is to do a test cross, and that's breeding the mystery individual with someone who's homozygous recessive, and that way you can figure out what's going to happen. So if any of the offspring display the recessive phenotype, then the mystery parent has to be heterozygous. So here we've done our test cross. We have, well, if we are not sure if it's homozygous dominant or homozygous recessive, but we have bred it with a homozygous recessive. I'm sorry, we don't know if it's homozygous dominant or heterozygous. We'll make it HT. We breed it with a homozygous recessive individual. If it was homozygous dominant, then you'll never, ever, ever, ever have any white offspring. But if it was recessive, if it was heterozygous, then you're going to have some recessive traits in there, right? Because dad can only give you the recessive trait. Mom, we're not so sure about. But when you breed them together, if you get recessive traits, then you know it's got to be heterozygous. Has to be, because it had to be able to provide that recessive trait. All right, so another thing that we talked about this, that Mendel derived that law of segregation by looking at a single character. We also have the law of independent assortment. And this is that when Mendel did his project and he was looking just at pea color, we were looking at that one character, those were called monohybrids. He was looking at only one single character. When we cross between like heterozygotes, we call it a monohybrid cross. When we were doing all these experiments before and looking at these Punnett squares, it was a monohybrid cross. We were looking at only one character. Mono means one, single. But let's say that we want to look at two characteristics at the same time. This gets a little bit more complicated, but we call this a dihybrid cross. It's a dihybrid cross between F1 dihybrids can determine whether two characters are transmitted to offspring as a package or as independently. And what he found was that the law of independent assortment, it isn't connected to one another. And when you have a dihybrid cross, your phenotypic ratio is now 9 to 3 to 3 to 1. It's 3 to 1 when it's a monohybrid cross. When it's a dihybrid, it's 9 to 3 to 3 to 1. It's the number, if you add up 9, 3, 3, and 1, it's the number of squares within this dihybrid cross, within this Punnett square. So this should be, y'all will be doing this in lab to help this make a little bit more sense. I would really, really strongly recommend that you do some practices on it online. There's lots of resources for you to do it. It's very difficult to do this in a recorded lecture when we're not sitting face to face. That's one of the things that I don't love so much about doing online classes. It's a lot easier when we're face to face and we can play this out on a chalkboard. So when Mendel did his dihybrid crosses, that was the basis of his law of independent assortment. It's that each pair of alleles segregates independently of any other pair during gamete formation. So color has nothing to do with the wrinkliness or smoothness of the peas. This law applies to genes on chromosomes that are not homologous or those far apart on the same chromosome. So genes located near each other tend to be inherited together. Basically know that genes separate independently. They're not linked. They don't hold hands and pull each other over. It's the law of independent assortment. And it's the dihybrid cross that proved that. Probability laws govern Mendelian inheritance. So Mendel's law of segregation and independent assortment reflect the rules of probability. The outcome of one coin toss has no impact on the outcome of the next one, right? So you go to Vegas and you play the roulette table and you look and you see, oh my gosh, it's landed on black 12 times. And so you put all your money down on red. It has the exact same chance of landing on black again or even green. Just because it's landed 20,000 times on one color does not mean it has to land on a different color the next time, right? It's in the roll of the dice or the roll of that wheel is independent of what happened prior to it. So in that same way, the alleles of one gene segregate into gametes independently of another gene's alleles. So one gene moves off and it doesn't matter what the other genes do. Like I said, they don't hold hands. The multiplication rule states that the probability of two or more independent events will occur together is the product of their individual probabilities. So you can look at this on a monohybrid cross. Segregation in a heterozygous plant is like flipping a coin. Each gamete has half a chance of carrying the dominant allele and half a chance of carrying the recessive allele. It's just a flip of a coin because you only have, it's a 1 in 2 chance, right? 50-50 chance whether you get the recessive or dominant allele if your parent was heterozygous. So here you can see it with the with the example of looking at flipping a coin. But ultimately, they're independent of one another. It doesn't matter what happened before. The addition rule states that the probability that any one or two or more mutually exclusive events will occur is calculated by adding together their individual properties. So it can be used to figure out the probability of an F2 plant from a monohybrid cross will be heterozygous rather than homozygous. We can apply the rules of probability to predict the outcome of crosses involving multiple characters. It's kind of fun to do if you figure out and you learn what, you can just Google it and find things out. But let's say that you figure out that you exhibit a certain trait and your partner exhibits a certain trait. You can play with Punnett square and you can come up with some sort of a prediction to determine what is the likelihood, what's the probability that your offspring together will exhibit one of the traits of the both of you. A dihybrid or other multi-character crosses is equivalent to 2 or more independent monohybrid crosses occurring simultaneously. In calculating the chances for various genotypes, each character is considered separately, and then the individual probabilities are multiplied. So when we looked at a dihybrid cross, it's looking at 2 traits at one time. Like, what's the chance that you are yellow and smooth, or green and smooth, or yellow and wrinkled, or if you don't care about that, you could just look at yellow and green, and you could just look at wrinkled and smooth. So that's what that's saying, is that you can do them independently, or you can link them together. Okay, so on this one, is this dihybrid? I see 1, 2, and I see 3 traits here. So this is actually a trihybrid cross. And we're not even y'all aren't gonna even have to deal with that. If you take genetics, you'll get that far into it. But for us, we're gonna keep it simple with dihybrid. And as much as I possibly can, I'll just keep it with monohybrid. If you can just remember that a dihybrid parent, if the P1 generation is true breeding, or if the parents are for sure heterozygous, you will always have a 1-to-3 ratio if it's a monohybrid cross. If it's dihybrid and the parents are both heterozygous for both of the traits, it's always a 9-to-3 to 3-to-1 ratio. The answer is given there for you. You don't even have to do the Punnett square. It's known. Not all heritable characters are determined as simply as the traits that Mendel studied, but to get started with this concept, that's the easiest way to look at it. But the basic principles of segregation and independent assortment apply to even more complex patterns of inheritance anyway. So he looked at very simple things. We're going to go into how things are more complex. The same laws still apply. So let's extend Mendelian genetics for a single gene. Inheritance of characters by a single gene may deviate from simple Mendelian patterns in the following situations. Sometimes alleles are not completely dominant or not completely recessive. And we'll talk about what this is called. It's kind of called codominance, where maybe a purple flower and a white flower could produce purple, white, or pink. You get a little mix of it. Or when a gene has more than 2 alleles. So in the case of like skin color, we have multiple alleles, multiple alleles, or even height that make up how tall we become. And it all, you could either get loaded up with the tall genes or loaded up with the short genes, or somewhere in between. And that's why human height is so varying, right? Or when a single gene produces multiple phenotypes. So we sometimes with this first one, alleles are not completely dominant or recessive. These different degrees of dominance. So there's complete dominance, and that occurs when phenotypes of the heterozygous and the dominant homozygote are identical. We saw that with the purple flowers, right? There's incomplete dominance, where there's somewhere in between the 2 phenotypes somewhere in between the 2 parents. Then that could be in the case of the purple. Purple, white make a pink. Or there's codominance, where 2 alleles affect the phenotype in separate and distinguishable ways. So you could have, so here we have, I was saying purple, but let's do red and white for this flower. You get a little bit of incomplete dominance here because you can have red, pink, and you can have white. The relationship between dominance and phenotype. Alleles are simple variations of a gene's nucleotide sequence, because we have a very complex sequence of nucleotides, right? Our genes are made up of billions of different alleles. Allele is just one little tiny part of it to make it our brain be able to wrap around it. When a dominant allele coexists with the recessive allele, they don't interact at all, right? They shouldn't interact. They're totally separate from one another. You got moms, you got dads. For any character, dominant and recessive relationships of alleles depend on the level at which we examine the phenotype. So in the example of this disease called Tay-Sachs, it's a fatal disease. It's like an enzyme that you get too much lipids in the brain and the brain cannot function because of it. So at the organismal level, the allele is recessive. At the biochemical level, the phenotype is the enzyme activity is incompletely dominant. And at the molecular level, the alleles are codominant. So there's different ways that you can, I guess what this is saying is there's different ways that you examine the phenotype. Are you looking at the way the organism looks? Are you looking at the way the enzyme activity happens inside the body? Or are you looking at the molecules? So it can vary. Sometimes genetics aren't quite so simple. Frequency of dominant alleles. So dominant alleles are not necessarily more common in populations than recessive alleles, and we actually see this all the time. And one of the reasons why dominant alleles may not necessarily be more common is if it's a detriment to have a dominant allele. Eventually, the dominant allele will get out of the population. So, like one baby out of 400 is born with extra fingers. That's called polydactyly, but that's a dominant trait. Most genes exist in populations in more than 2 allelic forms. So the best example is looking at our blood group. There's 3 different alleles for, like if you have ABO blood, the ABO blood group in human is determined by 3 different alleles. I, dominant enzyme A, dominant enzyme B, and then the recessive enzyme. And it's a combination of these 3 that tell us if we're A, B, O, or AB blood type. We're not even going to get more into depth on this, just understand that sometimes it's more than one. It might be 3 alleles that determine the way that we are. Most of our genes actually have like multipliable effects on our body, and we call that pleiotropy. So pleiotropy alleles are responsible for multiple symptoms of certain hereditary diseases like cystic fibrosis and sickle cell disease. Some traits may be determined by 2 or more genes. The gene products may interact. Alternatively, multiple genes could independently affect a single trait. I'm trying to get into some pictures of this so it can help understand it. So epistasis is a gene. A gene at one locus alters the phenotypic expression of a gene at a second locus. So Labrador retrievers and a lot of other mammals, coat color actually depends on 2 genes. So you know that labs they're either black chocolate or yellow. I know there's people that say they have white labs. I know they say silver. These are all just variations of yellow, of chocolate, right, or of black. White, red, silver. Silver is a type of chocolate, and yellow labs can sometimes be white in color or more red, but it's 3 specific colors that they are, and then variations within that. One gene determines the pigment color, whether they're dark or light, and then another gene determines whether the pigment is actually deposited on the hair. So when you look at it, there's many options that. So if you say you take 2 black labs, you have dark or light, or black or brown, big B, little b, and then whether the pigment is actually given to the animal. And if the pigment is not placed onto the fur color, then that means that you've got yourself a yellow lab, right? So multiple alleles affecting different things in kind of a really funny way. Quantitative characters are those that vary in a population along a continuum. So we call this polygenic inheritance, and that's where you kind of have an additive effect of more genes. And skin color is the primo example of that. So to be in a light skin color. So basically, there's multiple, and this is simplifying it, there's probably 50 genes that determine your skin color. But all of these, you could get your dad is really, really dark skin and your mom's super duper light skin, and you're going to probably fall somewhere in the middle. But let's say that you see a family where dad is really dark skin and mom is really light skin, and they're crazy and they have 10 kids. There's going to be a huge range among these children. And we see that a lot of times, like if two African American, let's call them the the P generation, if they, or actually, you can see this when you have an African American person and a really, really Caucasian white person, right? They have an offspring that's somewhere in the middle, right? Not super dark, not super light. If that individual has children with another individual just like them, the range of color among those children can vary. They can be as dark, dark, dark as the dark African American grandparent was, or they can be white, 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 just like really light skinned Caucasian, because the genetics, all those alleles in the two parents vary so greatly. If you have two parents that are really dark skinned, most likely they're going to have children that are a little bit dark skinned too, or that are similar in color to them as well, because they're passing on similar genes to them. Same thing we see with height. Multiple things are feeding into it, but also nutrition and other other outside variables affect that as well. Another departure from Mendelian genetics arises when a phenotype for a character depends on environment as well as genotype. The norm of reaction is the phenotypic range of a genotype influenced by the environment. So nature versus nurture, there is some environmental impact on the way that we look. Height would be an example of that. You could have not had proper nutrition when you're younger. You could have suffered from some sort of a disease that stunted your growth. You could have not been exposed to the proper type of vitamin or mineral, and that caused some issue with you. You could have had cancer as a child, and the chemotherapy caused issues in your height. Sometimes just body shape is genetic, right? Whether you're naturally kind of thin or whether you're naturally more muscular, whether you work out, whether you eat right, all those things are going to have some kind of influence as well. So nature or your genetics, and nurture is what you do to them. That has a major impact on our phenotype as well. The phenotypic range is generally broadest for polygenic characters. So such characters are called multifactorial because genetic and environmental factors collectively influence our phenotype. Mendelian view of heredity and variation. An organism's phenotype includes its physical appearance, internal anatomy, physiology, and behavior. An organism's phenotype reflects its overall genotype and unique environmental history. Many human traits follow Mendelian patterns of inheritance. So humans are not really great subjects for genetic research because it takes a really long time for us to have a child, and then for that child to be ready to produce offspring itself. And so usually if you're a scientist, you might get through one or two generations and before you're dead, before you die off yourself. And so we don't use humans to study genetics because it's way too hard to do it. But basic Mendelian genetics endures as the foundation for our human genetics. And the only way that we can really do this by studying multiple generations is looking at pedigrees, understanding the genetics of individuals based on their phenotype many, many generations prior to your generation. So a pedigree is basically a family tree, and it describes interrelationships of parents and children across generations. Inheritance patterns of a particular trait can be traced using these pedigrees, and we can use it to make predictions of future offspring. So do not be surprised if you see on the exam a complex, and don't let it overwhelm you with how big it is. But you might see a pedigree chart, and you might have to determine whether someone's heterozygous, whether they're homozygous recessive, or give a prediction of what certain offspring are going to be. So we're using this trait of widow's peak here. And then we're also looking at, I wish it didn't do this, but, oh, I'm sorry. So there's two separate, looks like there's two separate pedigree charts happening inside here. Let's just look at widow's peak. So you have your grandparents. So widow's peak is, I already told you the answer to this, but is widow's peak dominant or is it recessive? Well, if we look here, this girl has widow's peak. She's either homozygous dominant or heterozygous. So obviously the answer is that it's a dominant trait. So our first generation grandparents, grandpa has widow's peak, grandma does not. Over here, your other set of grandparents, grandpa does not, grandma does. And so when they have children, that's what this line represents. They now have children. They had two females. These guys had two females. These grandparents had 4 kids. Here's your dad, and here's your mom. So that's mom's parents, and this is dad's parents on the left. You have you got 2 sisters that are born out of your parents' offspring or union, and that's what this line. So the 2 people connected vertically mean that they were married or produce offspring, and then the down line shows you who their offspring are. So mom and dad are heterozygous. You produced one sibling that had no widow's peak and then another sibling that did. And we don't know until this sibling has more babies themselves with somebody who maybe is or isn't with widow's peak, whether that sibling is homozygous dominant or heterozygous. Let's say that this girl had a child with a man who had widow's peak, who has widow's peak, and their child has no widow's peak. What does that tell you about both of the parents? What that tells me is that they both have to be heterozygous because they had to pass on the recessive trait to that child, right? It's actually the same exact situation as you see here with this child for offspring. The fact that she has no widow's peak at all tells you that the parents have to be heterozygous because they have to carry the recessive trait. In this case, they're doing that PTC trait thing. It's like a chemical, and I'm sure y'all have tried it before, and you might actually do it in lab. But it either doesn't taste like anything, or it tastes terrible, 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 like real, real super bitter. And it's actually a genetic trait. So let's try to figure out if you can taste PTC, is it dominant or is it recessive? Well, so you have, so if it's colored, you have the trait. So you have mom's or dad's parents who can't taste it, and on mom's side, you have grandpa who can. Grandpa gives his recessive trait to mom, right? And then dad ended up heterozygous. So we have 2 heterozygous parents who had 2 kids. One can't taste it at all, and one can. And we don't know whether she is homozygous, dominant, or she's heterozygous. But again, we can find out because if she has a child with a man who can taste PTC and that child cannot, then we can assume that she and her, the mate of that child, are heterozygous. You might want to rewind this portion and listen to it a couple times until it seems to make sense. Do some practicing on this one because I know that it's very complicated until it becomes simple for you. And I hope that that happens. But this is, again, Mendelian genetics is a lot of fun, I think, to play with and do different experiments with, but it can be confusing. And with an online class, it's very much self-learn, no matter how hard you try to sit through these online lectures and other things. You still have to go back and teach yourself because I'm not able to do things on the board and answer your questions, etc. So make sure that you do as much practice as you possibly can on these pedigree charts and on the Punnett squared. So sometimes you have recessively inherited disorders. So many genetic disorders are inherited in a recessive manner. And so you could go many, many, many years without anybody in your family having a disease, and then it just randomly will pop up. And that's because you've been carrying this recessive gene through the generations until you finally had a mate who had it as well. And you had that 1 in 4 chance of passing it on to your offspring who's born with it. Sometimes also the range is mild to life-threatening. So if you are a heterozygous individual, we call you a carrier. You're carrying that recessive allele along, but phenotypically you don't show the disease, but you're carrying it. Most people who have recessive disorders are born to parents who are also carriers of the disorder, who are carriers of the disorder. So like in the case of albinism, if you're albino, both of your parents had to have carried that genetic disorder. They had to have carried that gene. Most of your offspring are probably, right, if you have 4, you have only a 1/4 of a chance to have a child born with albinism, but it's a 1/4 chance every time you have a child. So could it happen where you have 2 out of your 5 kids? Absolutely. Remember, it's a flip of a coin. It doesn't matter. If a recessive allele that causes the disease is rare, then the chance of 2 carriers meeting and mating is pretty low. However, when you have like close relatives mating with one another, the chance of that occurring is a lot higher. So most societies and cultures actually have laws, and if there are no laws, then it's definitely taboo, culturally taboo, to have marriages between close relatives. That's why in the old days, royalty generally had the same diseases. They died young because they were passing on the same terrible genetic disorders from one offspring to the other because there wasn't good genetics, right? You weren't able to bring in kind of some fresh and better genes. You were just ultimately narrowing it down so that recessive disorders were showing up more and more and more often. So cystic fibrosis is it's a genetic disease, pretty common in the U.S. actually, and it strikes 1 out of every about 2,500 people, mainly of European descent. And it's we don't need to really get into it, but basically what happens is that mucus builds up, and it can become mucus builds up in the organism, in the organs. You hear about it with the lungs, and but it's in other things. It absorbs, so it steals the nutrients from your body, and it can be life-threatening many, many times. So that's an example of one. Sickle cell anemia, sickle cell disease, is an interesting disease that affects 1 out of 400 African Americans. So that's pretty common. And let me see if I can get to a picture of it. But it's sickle cell anemia is a homozygous recessive disease, and in Africa, if you have sickle cell anemia, you usually don't live very long. It's a lot of physical weakness, kind of like an autoimmune disorder almost, where your body just isn't working properly. So you can cause stroke and paralysis, lots of organ damage, extremely, extremely painful. So you might ask yourself, like, why then does it stick around, right? So if you're heterozygous, they're said to be usually healthy, but might every now and then suffer from some symptoms. And so why does this terrible, terrible disease that is so debilitating to people, why is it still around? Why wouldn't eventually evolution and natural selection wipe it out of the gene pool? And the reason why it doesn't is that if you're heterozygous, since this is African Americans living in Africa, where malaria is super duper duper, or if you are a true African living in Africa, malaria is super duper duper common. much, much, much more deadly actually than sickle cell anemia is. If you have sickle cell anemia, or if you're heterozygous for sickle cell anemia, you're less susceptible to getting the malaria parasite because it can't get into your blood because some of your blood cells are shaped a little bit odd. So there's actually an advantage to being heterozygous, which is why it's still in the gene pool. So it's a terrible disease, but there is somewhat of a benefit to having it. So this is kind of, your red blood cells get this sickle shape and they can't carry oxygen. It's very debilitating. It's a terrible disease to have. You wouldn't wish it upon anybody. But if you're heterozygous, your sickle cell trait is somewhere in between a healthy blood cell and a sickle cell blood cell, and so you don't get malaria. So there's a benefit to it. There's a benefit at least to being heterozygous. So that's why it stays in the gene pool. Some human disorders are caused by dominant alleles. So dominant alleles that cause a lethal disease are rare, and it usually arises by some sort of a mutation. Achondroplasia, it's a form of dwarfism, is caused by a rare dominant allele. So you can see here, like you might have had a dwarf dad, but a normal mom. Or I'm sorry, your parents. Yeah, you had the P1 parents were a dwarf, mom was normal height, and so dad was heterozygous. He gave the dwarf allele here. Mom can only give the normal allele. So if you look right here, what's the chance? It's a 1-to-1 ratio here. It's a 50-50 chance. So when you have parents who have the dwarfism gene, or at least this specific one, if one of the parents is normal sized and the other one is not, then it's a 50-50 chance that that child will be normal height or dwarf. So this will end chapter 11. Like I said, make sure that you really spend some time doing some practices, looking through the book, understanding the Punnett squared in a pedigree chart. You're going to see questions like this on the quiz, on the homework, and on the exam. And it can be kind of complicated. It can be especially complicated because we don't meet in class to really discuss this stuff. So if you have questions, make sure that you reach out to me. If you want to, you can ask your lab instructor. You can also go to the Bird Sanctuary Tutoring Lab to get information about this. There's tons of resources for you, so make sure that you utilize them if you're having difficulty, because it can be something that's difficult to comprehend, but it's definitely comprehensible.